Today's presentation is from Dr. Sandy Lau. She's a postdoctoral researcher from the Department of Obstetrics and Gynecology at the University of Auckland. Sandy, please.
Thank you for your introduction. I wanted to share a paper today that is relevant to me in my field, looking at hypertension in pregnancy. This paper is Circulating Extracellular Vesicles and Normotension Restrain Vasodilation and Resistance Arteries. Although it's not directly relevant to what I'm studying preeclampsia, however, it's looking at the effect of EVs and how they actually affect vessels, which is directly relevant. This paper was actually published in January in the Journal of Hypertension. And I chose this paper because I thought the authors did a really elegant job of separating the effects of EVs that they've isolated from plasma, and normally you wouldn't really know what's going on, and but they've done a really good job at trying to tease out what they can given the situation. So the main aims of their paper is actually to look at plasma EVs from normotensive and hypertensive rats and how they affect a particular response, the acetylcholine-mediated vasodilation and small resistance arteries. And in hypertension, the vascular resistance, 50% of it at least is contributed to the small resistance arteries. And then they tried to determine whether or not nitric oxide synthase is contributing to the observed effect. They then try and saw whether or not actually having an intact membrane of the EVs determines this effect. And then because this so far is not clinically relevant at all, you're looking at 2 different strains of rats. They then did a small amount of work looking at human plasma EVs from hypertensive and normotensive patients and whether or not they can actually repeat the findings in mice. So just a little bit of background, they use a spontaneously hypertensive rat, and these rats have been bred to develop hypertension, and that leads to cardiovascular diseases later in life. And the parent strain that the SHRs are derived from are the Wistakiotos, and they remain normotensive throughout their lifespan. And the SHR is a very valuable tool in physiology because the phenotype and the disease progression is very well characterized in the literature. And these animals start developing hypertension from 6 to 8 weeks old. So in this study, they have both 6-week hypertensive or pre-hypertensive rats, and they also look at the full-blown hypertensive rats at 12 weeks. So how they isolated EVs in the blood, this is more of a physiology-based paper, and it doesn't necessarily has as much characterization as you would expect from an EV biology paper. But what they did tell you was that they collected blood and citrate buffer. They spun that plasma the platelet poor plasma at 3,200 G for 10 minutes, took the supernatant, spun it at 17,000 G for 30 minutes, and then again the supernatant at 116,000 G for 75 minutes. And that's for the rat EVs. For the human EVs, they did a 20,000 G spin. And the first thing that they did was actually characterize the EVs that they got from the spontaneously hypertensive rats and the normotensive rats. So they looked at the morphology of the EVs using cryo-EM. and I really don't have expertise in the area, but the paper states that the EVs in here at the different magnification look similar, and I just took their word for it. They also looked at the size and the number of EVs using the Zeta view, and it all seems to be centered around the same size. And when they used the quantification, it doesn't seem that the numbers between animals at 6 weeks, whether it's normotensive or hypertensive, it doesn't seem to be statistically different. Then they performed Western blots to confirm what they're using are EVs and not non-EV protein fragments. So they used the tumor susceptibility gene 101 as a marker of an EV and calreticulin as a negative marker. And their control for these things were the microsome membrane preparations. So next they wanted to know, because it's actually plasma EVs and you don't know what is in them, where the EVs actually, what cells the EVs actually came from, they used the image stream X Mark II to perform some flow cytometry. Using the markers CD105 for endothelial cells, CD42 for platelet cells, leukocytes being CD45 and CD31 positive. They did, however, find that in hypertensive rats at 12 weeks old, so this is full-blown hypertension, that you get a significantly higher proportion of the total EVs being from a leukocyte derived, but the actual numbers of EVs weren't different. It's just the proportion that was different. So this is more sidestepping for a minute and just confirming the hypertensive phenotype of the animals they used and getting a baseline in the vessels to be able to compare what effect the plasma EVs have versus what effect is actually contributed by the phenotype of the animal. So as expected, the blood pressure of the SHR increases with age, but not in the normotensive animals. And to assess the contractile properties of these vessels, and that you still have, after isolating from the animals, you still have intact smooth muscle and endothelial cells in the blood vessel fragments that they used. They looked at phenylephrine and a vasodilator NS309. And the technique that they used in the rest of the studies is pressure myography. And one of the requirements of publication for this type of experiment is to be able to show that when you're taking these vessels for pressure myography, that you still have, in essence, alive vessels. So what's actually happening in this is that these vessels, 2 millimeter fragments, about 100 to 150 micrometers in size, were taken from the mesentery tissue surrounded by the gut, and they cannulate it to the pressure myograph. And what it does is it puts a pressure through the lumen of the vessel to mimic what you get normally in an animal. And it also allows buffer that you can pump through flow as you would have in blood flow. And there is a camera overhead which assesses the diameter of the vessels as you put different vasoactive drugs in the surrounding buffer bath. And that's what they use to determine the diameter and the percentage relaxation. So what they can see is that in hypertensive vessels, they actually don't contract as much as normotensive vessels do, and that could suggest that the vessels are already in a pro-constrictive state, and therefore there's only so much more it can constrict. But then when you put a vasodilator in there, both vessels were able to dilate between 80 to 100%, which means that throughout the process of obtaining and instrumenting these vessels, that they still have intact endothelial cells and intact smooth muscles. So so you can actually have more confidence in the results that are to follow. So they performed a dose response curve to acetylcholine. And the way that acetylcholine works is that it induces vasodilation by binding to endothelial cells. It then stimulates nitric oxide production via nitric oxide synthase. And this nitric oxide diffuses into the neighboring smooth muscles to cause relaxation. So if we looked at the panel C, actually, if we only looked at panel E, the dose response to acetylcholine in the hypertensive or the normotensive animals are the same. There's no difference there. However, if you looked at panel C and you apply an inhibitor of nitric oxide synthase, L-name, you can see that it blocks the response of these vessels to acetylcholine, especially at the higher concentrations. And it does that to a lesser extent than it does in hypertensive vessels. And that's actually, this is all expected. We are establishing a baseline in this sort of this slide. None of this is novel or remarkable in the field of hypertension. So once they have established the baseline of what they're actually comparing to, then they tested what effect plasma EVs has to the resistance vessels. And they took the plasma from the animals, isolated EVs from the same animals as the small arteries used, and then adding the EVs both into the bath that it is in, as well as adding it inside the lumen of the vessels for 10 minutes. So for example, in panel A, using normotensive rat blood vessels, they have actually put the EVs back from the same animals that they put the same EVs as back into where the arteries came from. What's not clear is whether or not it was exactly animal match for each individual experiment, or whether it's swapped between groups, or whether they actually pulled the EVs and then put the pulled EVs in. And I can't, from experience, I can't see this actually being pulled EVs purely from the amount of work each experiment requires. Unless they froze down the EVs, it can't actually be done in that sort of time frame. And they didn't mention freezing and defrosting EVs. In the control, what it means is that they didn't add any EVs into the bath. So if you added normotensive EVs to normotensive arteries, you get a reduction in acetylcholine mediated vasodilation. If hypertensive EVs is added to normotensive vessels, so we're looking in panel C here now, you don't get the effect. There is no difference between adding EVs and not adding anything to the vessels. And that suggests that there is something on the normotensive EVs that may be lost on the hypertensive EVs. And the hypertensive vessels, so we're looking at this side now, neither adding hypertensive EVs, which is panel D, or normotensive EVs, panel B has any difference. So it doesn't matter if you add anything in or not add anything in, you're not getting that same restrained vasodilation. And this might suggest that not only is there something on the normotensive EVs, that restrained vasodilation, there is something on the hypertensive arteries, which means that they're not responsive to whatever that was on the normotensive EVs. And although remembering in the previous slide, we saw that there is no difference in the baseline response to vessels to acetylcholine. So this isn't just a different response of the vessels to acetylcholine between groups. It is actually, they've managed to separate it to say that it is a different in the effects of the EVs. So nitric oxide signaling is a large contributor to the control of vascular resistance and a mechanism by which ACH-mediated vasodilation occurs. And nitric oxide is present in EVs. So for example, in my area of study, placental EVs, it is present on placental EVs. It is functional, and actually the function is reduced in hypertensive disorders of pregnancy like preeclampsia. So the authors wanted to know whether the restraint and vasodilation is a response to the normotensive EVs is due to the reduction in nitric oxide signaling, or if blocking the signaling adds to that, meaning it occurs by a different mechanism than nitric oxide. So if we looked at panel A, which is normotensive EVs added to normotensive arteries, it shows that when you then further add a blocker of nitric oxide, that it doesn't result in a more significant restraint of vasodilation. And if we looked down here in panel E, in fact, you could get the same response by just adding the blocker and not the EVs itself, meaning the EVs aren't necessarily necessary. So what happens if you add hypertensive EVs to normotensive arteries? So in this figure, all of these are added to normotensive arteries. So adding hypertensive EVs. Now, in the previous slide, we didn't see an effect of hypertensive EVs on hypertensive vessels, but then if you add, or normotensive vessels, but then when you add the blocker of nitric oxide signaling, you rescue the lost effect of restraining vasodilation. And again, if you looked at the bottom, that if you added the blocker alone, the EVs sorry, my daughter's actually just come into the room. If you added the blocker alone, then EVs aren't necessarily needed for this effect. And what it's trying to say, what the authors are trying to say by doing these experiments, is that the normotensive EVs work by affecting the nitric oxide signaling mechanisms. And this is what's missing in the hypertensive EVs. So why does the SHR EVs not restrain vasodilation? Why is it different? And the funny thing is, going into the EV field, I would have expected that hypertensive EVs are pro-constrictive. So they'll have a, they'll restrain vasodilation even more than the normotensive EVs, rather than being the other way around, especially because, well, what the authors suggest is that these animals are actually suffering from chronic hypertension. I mean, this is 6 weeks after it's developing hypertension. And the author suggests that this is a compensatory response. And in some respect, if you change the perspective, it starts to make sense, because if you get vessels that are pro-constrictive and your EVs are pro-constrictive, well, the animal isn't going to survive for a very long time. So it does make sense that this could potentially be a compensatory mechanism. So the next thing the authors did was asked whether an intact EV lipid membrane is required for the effects seen. So they removed the lipid membrane from the EVs using chloroform methanol, and treating normotensive arteries with delipidated normotensive EVs produces similar effects as lipid intact EVs. However, when you treat normotensive arteries with delipidated hypertensive EVs, you have rescued the restrained vasodilation, so it behaves more like a normotensive EV. And the author interpret this as it is the cargo that the EVs, that is the required portion to exert this vasoconstrictive effect. And to rule out that it is the lipids itself that produces this effect, and it's not necessarily, you don't necessarily need the micro EVs, what they did was added an additional group where they put in liposomes, which didn't have the same effect, and they didn't actually show the raw data from that though. Okay, so the final question they asked is the full-blown hypertension phenotype required to produce this effect, or are the EVs altered even before the development of hypertension? So what they did was then compare 6-week-old pre-hypertensive EVs to 12-week-old hypertensive EVs. So what you're looking at should be, and C, D, and E should all be normotensive arteries. And unsurprisingly, there is no difference, but if we're looking on panel C, so normotensive EVs on normotensive arteries. And unsurprisingly, there's no difference in the acetylcholine dose response, because there's no change in hypertensive status, and therefore you expect no change in their EVs. However, if you looked at 6-week-old hypertensive EVs versus 12-week-old hypertensive EVs, so looking at pre-hypertension versus full-blown hypertension, the 6-week-old pre-hypertensive EVs behaved similarly to the normotensive EVs. And that is suggesting that, that with the EVs actually change with progressing development of hypertension. And this sort of supports their speculation that the loss of restrained vasodilation is a compensatory effect to counteract the hypertension in these animals. As these animals get more hypertensive, the EVs lose the ability to restrain vasodilation. And that's all great, but then I feel like this is probably at the point where they sent it to a reviewer and actually got the paper rejected, saying, hey, this is nice, but everything's been done using rats. The question is whether this is specific to the strain of these bred hypertensive rats. Does other rats also exhibit this, or is this a feature of hypertension across species? So what the authors did were to isolate plasma EVs from hypertensive and normotensive patients and look at the effect of mouse mesenteric arteries. Note that we're now changing from rats into the effect of human on mice. So we're involving 3 different species now. Again, they were able to show that normotensive EVs, so the black line, were able to restrain vasodilation compared to control, and that hypertensive EVs does not have this effect. However, I find that the differences, if you look at the scale bars, you know, you're talking about 100%, the maximum scale bar is now 60%. I find that this data is, the difference between hypertensive and normotensive EVs is much less convincing than the animal data. So maybe it does still have this phenomenon, and then it's the effects are dampened, or something else. It definitely needs to be reproduced for us to have confidence in it. So in quick summary, what these guys have shown is that in normotensive EVs, when you put normotensive EVs at 6 and 12 weeks, as well as normotensive delipidated EVs to blood vessels, you're able to restrain nitric oxide mediated vasodilation. And the same thing as when the hypertensive animals, before they develop the full-blown hypertension, they're also able to restrain it. When you've got full-blown hypertension, you're no longer able to restrain the ACH-mediated vasodilation. But then when you take these EVs, strip their lipid off, once again, they're able to restrain it. So the strengths and limitations to this work, well, I really liked that they're able to, their experimental design actually separated the effects of EVs from the underlying effects of vasculature and what hypertension actually does to the animals. But they also put interestingly, they put the same EVs back into the same animals. So from an immunological perspective, doing this in ex vivo and being able to in the future do that in vivo, it kind of is a positive, because you won't have all these questions about whether or not this is an immune effect, because low-grade inflammation is related to hypertension. But one of the main thing is that they're ex vivo experiments, and they're it's a very short time period that they've incubated for, only 10 minutes. And when they take the same vessel, the EVs from the same animal, put it back into the vessels, my question is, shouldn't there already be an effect? Why is there an additional effect? Unless and the time it takes for them to load the vessel and extract the EVs, that whatever effect the EVs had while in the animal is gone. I don't understand why they see an additional effect. Another thing that we need to appreciate is that ACH-mediated vasodilation is not the only controller of vascular resistance. There are a whole heap of other things that could be playing a role, or the EVs might have an effect on other controllers, such as the endothelin run system, the angiotensin system, or even it might even have an effect in the central control, the neural control of blood pressure. And actually, altered vascular resistance really isn't the same as altered blood pressure control. So even if you alter vascular resistance, EVs are able to do that, it doesn't directly translate to hypertension, because blood pressure homeostasis is integrative. So you can compensate for the effect of the EVs by changing your blood volume, or you could reduce the neuro input to reduce vascular tone, or you could be shifting blood between your arterial and venous circuit and pooling it in your venous circuit. So there is less volume in your arterial circuit to increase pressure. So there's all sorts of these other things that aren't actually answered. And therefore, the future work really that I assume that they're doing now is that you need to look at the biology of the EVs. What exactly are in the cargo of these EVs, and what else does it affect? And what I find that is really interesting is that the proportion of EVs have changed to a more leukocyte-derived proportion. And is it the leukocyte-derived ones that have the effect, or is it the endothelial-derived ones that has this effect? Maybe if they could somehow sort with the flow, then you could only put back the leukocyte-derived ones or the endothelial-derived ones. So there is plenty of research that can be conducted following on from this work, which has me really excited now. And not only placental EVs, but also endothelial-derived EVs. And that is all I have for today. If anyone has any questions.
Thank you very much, Sandy, for selecting this interesting paper and also for your analysis of this information. Any questions for Sandy?
Hi, Sandy.
Mm-hmm.
Yeah, I got a few questions. What volume they used for isolation the plasma EVs?
They said it's 12-week-old rats. They were able to obtain 7 to 8 mils of whole blood and 5 mils from 6 weeks, and that just reflects the amount of blood in these animals, because the 6 weeks are really small and blood volume is directly proportional to the weight of these animals.
So they used the 5 mil to spin down to get the EVs?
Yes.
Okay. They didn't do any dilution?
No.
Okay.
So for the actual myograph experiments, the amount of EVs they used, they normalized it to the amount that they got from 1 mil of blood. So even though they collected from 5 mils, they only put 1/5 of it back. But the proportion of so many EVs to the tiny, tiny size of these vessels, and you're talking about 7,000 square meters of endothelium, of blood vessel surface area, and then to take 2 millimeter segments and then throw in EVs from 1 mil of blood is really an overkill.
Yeah, and another question, if you can go back to the figure 1, is it possible?
Yes. So the characterization of the EVs?
Yes, that's in figure 1, yeah.
Let me just share my screen again.
Sorry.
Is that the one?
Yes. Let me have, my question is, the amount of EV between 6 week and 12 week in SHR, is the amount the same or not?
They say that there is no difference.
Okay.
They say that there is no difference. So
okay.
Yeah, it would be these two.
Yeah.
The only thing that might be different, but I guess it's because they have lower numbers and you're not getting enough statistical power, is that it looks like in normotensive animals, there should be a difference in the EVs, sorry, in the 6 and 12 week animals. The paper actually did have a huge table that actually showed exactly what yield they got from each animal, what the average was.
Okay.
But it was just too hard to present.
Yeah, I agree. Yeah, no, I was just worried about whether the blood pressure associated with EV production. 6 weeks is before hypertension and 12 weeks are hypertension. So whether once the males got a hypertension or rats, the EV production will increase. Looks or not. Yeah.
No, I'm looking at the actual table now, and the average of 6 week pre-hypertensive animals was 1.43 to the 10 to the 10 and 1.27 for 12 weeks.
Okay.
So there is definitely no
difference.
increase.
Okay. Yeah. Thank you.
Okay. Any other questions? Sandy, I have a question about the delipidated EVs. Does it mean that these are EVs without a membrane?
That is my understanding. Yeah, they've removed the membrane of the EVs. I'm not sure. Like, I also have questioned that. I'm not sure. Does it still have its shape? Like, if you're, or would it just be actually soluble protein, which was inside?
How, in this case, if they don't have a membrane, how do they maintain their cargo? And
Cargo, yeah, it is.
yeah. And they showed that there was effect based because of the cargo. And yeah, I'm just wondering what cargo was and how it was delivered or maintained safely without a membrane.
Yeah, I am not too sure. I imagine that if it had a membrane, then the whatever dangerous things inside, I'm assuming there are dangerous things inside a hypertensive EV, that would be bound and will never react with any surface receptors. Whereas if you just had the cargo content and you just expose the surface receptors of the endothelial cells to that I'm surprised that itself doesn't produce an unwanted effect. I'm really not sure about that either.
Any other questions? And we don't have anything in the chat. Thank you very much, Sandy. It was a very interesting paper for today. Thank you.
Thank you.
Thank you.
Guys, based on today's poll, so the preferred frequency of our meetings would be once in 2 weeks. So that means that we don't have a journal club next Thursday, and we will meet again on 4th of June. Thank you very much for joining us today. The recorded video will be on our YouTube channel, and I will send invite and information about the next speaker probably next week. Have a good weekend, and we will see you in 2 weeks.
Thank you.
Thank you for joining. Bye.